I use this slides show the dynamic interaction of rice and the pathogen determines disease occurs. So when the pathogen population contain a certain AVR genes, such as AVR-PIZT or AVR-PIA, they are recognized. This resistance activation leads to resistance cost. The resistance cost is defined as high resistance. So the next question is, we perform comparative RNA-seq between the susceptible LTH and the resistance lines. I show here 4 resistance lines, particularly the one line we call the RH, YH2115. This is the light cultivar overcoming resistance cost. And we found a cluster of genes in subcluster 3 and that's enriched in proteasomes. This gene encoding proteasome maturation factor UMP1, and this gene is sensitive 39 background, and we found the 26 proteasome bound and the activity all increased, leading to degradation of reactive oxygen species scavenging enzymes such as catalase, and which return leading to high accumulation of hydrogen peroxide in the line expressing these UMP1 genes. So I use this model to show UMP1 R2150 that confer enhanced resistance by promoting increase the 26 proteasome abundance activity, leading to degradation of OS scavenging enzymes, resulting. So this defense can cause resistance to multiple disease. And we found that indeed this cause broader spectrum resistance to rice plastic. We checked quite a few different strains, and they enhance the resistance to yeast blight and the bacterial blight and the rice fusarium moth disease. But the other traits have a low difference between the wild type and the lines carrying this near. So we can make a conclusion that elite varieties use special immune regulators to overcome resistance costs, such as UMP1 or 2115. And the next question is, because microRNA fine-tune various biological processes, which microRNAs involved in rice immunity against the Mycorrhiza aurelia? And to address this question, we examined the small RNA data between the susceptible line and the resistance line, and we identified a group of microRNA that differentially responsive to the infection of the blast fungi. And so far, in the past 13 years, 25 microRNAs reported to act in rice resistance to blast disease. And the left side, I show the published journal of these microRNAs. If you have interest in, you can check back to this journal and find the detailed information. Here I just show so many of the microRNAs are associated with ARGO1. So ARGO1. So this some module here I should highlight it in green also acts in other traits. And here I just show microRNA 168. This microRNA targeted ARGO1. So we constructed the overpressing and the target mimicry line, which leading to abolish of ARGO1 protein. And this module also regulates plant architecture and the other traits and the flowering time. Here I show the plants and the panicle and the statistical analysis data. Indicates this module can simultaneously improve blast resistance and the other traits. Because ARGO1 is a module impacts the other microRNA module, the answer is yes. We identified a group of microRNAs that's altered in the altered ARGO1 background. So this microRNA where I show as 4 here. So one is this 1320. this has a epigenetic effects to MR168 in prostate disease. And another one is 535. This one has additive effects to particle size. Another one is 164. So it also has additive effects on 168 in prostate disease. So this microRNA also regulates plant architecture and the flowering time as shown here. So the higher expression level of the microRNA leading to delayed flowering and the changing the shortened plant age. So summarize to microRNA module. This microRNA 168 targeted AGA1 associated with 3 microRNA that regulates resistance, particle size, and particle number, and a flowering time. So AGA1 is important for coordinates disease resistance and the growth. So AGA1 and its associated microRNAs coordinate immunity with growth to overcome resistance in media, but from first multiple with a leaving ovary, and raise immunity against this pathogen mainly occur at the lemma and part of the lemma poly, and the defense-related genes also induced only in lemma and poly. We found the application at the molecular plants for detail, and we found that higher expression of this gene correlates with resistance. So all the resistance lines show a higher expression of this gene to lower expression of this gene. So the higher expression and a higher resistance, but lower expression and lower disease resistance because the insertion. So finally, we use these genes and reach a elite cultivar we call the Yahui 2115. It's from this line. There are like 27 hybrid rice cultivars already released based on this one. And one overcome the among 10 backbone type of cultivars and one compromising cultivars. And this restorer lines carry multiple blast risk genes such as PI2 and PID2. And PI2 has a major contribution for prostate affair. And the cultivar we call the Yixiangyu 2115 obtained National Harvest Award. And based on this line, we obtained a targeted improvement of like flower time and also grain size, and we released more cultivars from this line, this bacteria, this germplasm. So based on this, we established a strategy for raising resistant cultivars. I don't want to go detail about this, but I want to say that the white wall grass and the parasitic wasps to control insects, and the flowering plants refugee parasitic wasps. So this is really ecological control for disease and the insects. So finally, take home message. Dynamic interaction of rice with a parasitic fungi determines the occurrence of the parasitic disease, and the loss of or variation of variance factors in the pathogen break down the resistance of rice plants carrying the corresponding R genes. And we introduced the one unique genes we call the UNP1 that's to overcome the disease custody, and the other one also resources for overcoming risk custody, and the higher expression of RAC1 leading to resistance to rice phosphamates. And I acknowledge my colleagues and the collaborators.
So our next keynote speaker is Professor Dr. Shah Alam Khan. He's working as a Dean Faculty of Crop Protection Sciences, University of Agriculture, Peshawar, Khyber Pakhtunkhwa, Pakistan. Doctor has concluded various research projects in Kansas State University, USA. Let us give a warm welcome to Professor Dr. Shah Alam Khan for his presentation.
Auzubillahiminash-shaytanir-rajim. Bismillahir-rahmanir-rahim. Welcome to the Honorable Foreign Guest, National, International, Honorable Deans, Director, Chairman, ladies and gentlemen. Thank you very much. Before coming to the podium, I saw that the State Secretary had given some chit to the earlier speaker to be brief. And this is the issue. Even at international national level, we don't have time beyond 12 minutes or 15. And I have seen in Europe and USA, same everywhere. Speakers are bound to 10-minute talks, so it is very difficult to conclude your talk unless you have a short paper. But anyhow, thank you very much. My talk is regarding the latest IPM tools. Dr. Khan. Before starting my lecture, let them sit on the. I would like to narrate a short proverb with you. A person came to the stage and he said, "I have been blessed with two super quality by the God." So the one quality was, he said, "I have a super memory. I have a super memory." And the second, he said, "I forgot." Got my point? You will laugh after 15 seconds. Yes? So the lecture is probably somewhere missing. Let him set it. Biochemical defense response of various rhizome plasm. Okay. Leaf and rice leaf folder, nephrocrosis medanalis. The theme and objective is important. Exploring tools of plant secondary metabolite as IPM tool and minimized pesticide used. Inclusion of HPR as an integral tool of IPM against insect pest. As IPM by himself, it embedded all the tools, but before we are using pesticide as an integral tool of IPM, what I suggest we should inculcate HPR tool in IPM as an integral part. Because we know that I have seen somewhere on the poster, the conference motto is sustainable solution for plant health. Very much important, very positive poster. But I would like to add for plant health to keep humanity healthy. Because sometimes the plant is healthy, but humanity cannot be kept healthy. As you see in the cities, in the market, in the vegetable fruit market, mostly I say the most fresh vegetable means high toxicated vegetable. In our part of the country, you see the salad, the cauliflower. So this is why we need to keep plant healthy to keep the human healthy. So natural plant defense system consists of phenomic, genomic, and proteomic approaches. But the two, genomic and proteomic, is a bit costly. It is not in the control of individual or farmer community or progressive farmer. You know, so far we are conducting and we have limited this type of research to our labs and having big projects. But the most cheap one, which I recommend for generally farmer community in third world country, that is the phenotypically. So if we know phenotypic mechanism of resistance, even the Dr. Penton was professor in 1925 in US. He is called as father of the resistance. So in 1920, wheat, Hessian fly, he said, and in latest technology in 2025, we just confirm that second stage larva is highly virulent. What I mean, that this phenotypic screening, if we do it as per laid down protocol, it can give you the endpoint like anti-zoonoses and antibiotics and tolerance. In anti-zoonosis, we can conduct it within 24 hours. Even 100 of genotype I have tested on a small space like half of the table. You just put the seed in a room with minimum 10 replication and release of in between the two rows as much as insect you want to test the plant against the insect. Within 12 hours, the insect which accept the plant will move toward the plant, and those who never prefer or non-preference type of insect, they move to the alternate host or stay stagnant. So the latest technology agreed that the anti-zoonosis type of characteristic, say the biochemical deterrents are there in the plant as a plant secondary metabolites, PSM. Most of the PSM, I will share with you 3 PSM plant metabolite we have estimated through proteomic assay. But the anti-zoonosis related plant secondary metabolites, they keep the pest away from the source, from the host. Physically, they have some trichomes, maybe shape, size, and color not acceptable to the pest. And biochemically, they have some odor, smell, which keep the pest away from the source. So based on this is called a speed screening of genotypes. We can do it with zero cost. Within the available facilities, St. University Tandojam in entomological department, plant protection, plant pathology. The second one is antibiosis technique. If we test the insect against the host, so feeding the insect and resultantly breeding the insect to see whether unsusceptible variety, they will give you clear population range, progeny production unsusceptible variety compared to resistant variety. I was testing soybean, rice, maize, canola. Within 12 hours, the susceptible plant, the seedling, attracted 15, 20, 30, 35 insects, while the resistant plant in 24 hours, maximum 2 to 3 insects would go there. So these type of techniques are very cheap, free of cost. Just we need to know the mechanism of resistance of different crops, screening against aphid, white fly, in virus cases like leaf curl virus, the yellow barley dwarf virus, because that is caused by Aerophilocephum pedi, and white fly in cotton leaf virus is caused by white fly. So these type of insect pests can easily be tested with this phenotypic screening on a small scale in every laboratory. Plant secondary metabolites play a pivotal role in mediating plant-insect interaction by influencing insect herbivore resistance through various mechanisms. So the diversity of secondary metabolites like alkaloid, terpenoid, phenol, glycosinolate, phenylalanine, ammonia-lyase, polyphenol oxidase, tannin, and foliar crude protein contribute significantly to plant ability to deter herbivore, attract natural enemy of herbivores, affecting herbivore behavior, growth, and development. Here this thing that the natural enemy is encouraged towards certain host. Again, the latest biotechnology technique prove that those host plant which are calling insect toward the source, they are releasing some volatile. A volatile compound is released by the plant, and resultantly number of insect pests are attracted toward the host. So volatile related compound means these type of compound are available in tolerant plant, because tolerant plant sustain maximum number of insects, and despite of feeding they will give us tangible yield. Compared to resistant plant are susceptible, tolerant resistant variety will give you tangible yield despite of the feeding. So this is another technique which confirm that certain volatile are released by the plant which call predator, parasite, and sometimes pollinators. This was the title of small paper as usual. One of my PhD student, Khaja Junaid, who have conducted latest research on rice cultivar. Importance of rice, all of us are aware of the rice, important. Almost 90% of the world is dependent on rice as a food we are using. Despite of many insect pests, one of the pests is leaf folder complex. Yes, coming to the complex. The rice leaf folder is a complex insect which folds the leaf once it enters and get shelter and enjoying inside the pulp of the leaf, the cell shape. This is a little bit history of the eggs, caterpillar, pupa, moth, and second stage larva of the leaf folder is highly virulent and destructive. These are the picture we have collected from the field. Plant culture and insect culture, we all know this type of mechanism and culture we can maintain on susceptible variety, the number of insects we need. Total 35 genotypes were tested at two stages of the plant. One was the vegetative stage and the other was the grain feeding stage. These 4 different enzymes were estimated and detected during our experiment. Phenylalanine ammonia lyase, which play important role in biosynthesis of the salicylic acid and which is the basic component of resistance signal system in plant. Second enzyme was detected as polyphenol oxidase. This also play a vital role in defense mechanism and disrupting its feeding ability, development and growth of the feeding insect pest. The third one, phenol, was also detected and estimated. It have the ability to bind with the dietary protein and its antioxidative nature, make it an important component of resistance by inhibiting the growth and development of the herbivory. Fourth one was tinine. This have also deleterious effect on by herbivore pest and have moth-mocking astringent properties. And the fifth one was unique. When the protein was detected and estimated, the higher amount of protein content resulted in increased intrinsic rate of increase and high population density of the herbivory. So at this stage, the protein estimation gave us otherwise result. The more we provide protein to the crops, the more the crop will be vigor, strong, but the more the crop will be appealing for the insect pest. So in this case, what I suggest, the farmer community should use appropriate amount of fertilizer for any crop under field situation. The limit they cross overdoses, a little bit dose can destroy the crop by the insect pest because it is more acceptable, more interesting for the pest. And these are the different test and table result where the stars show the resistant variety. When the crop rice plant was fed by the rice leaf folder, the more the plant was suppressed, the more the secondary metabolite was expressed. So they have almost following the Newton law. Action and reaction, but opposite in direction. So in case of protein, only it was otherwise. The rest, the resistant plant gave very good response. The more we fed the plant on the insect rice leaf folder, the more the resistant plant expressed against the pressure of feeding. So it means that this metabolite PL concentration have a significant impact in pest management. This is another PL concentration. The PPO concentration was also tested. Tinine concentration, total phenol content were estimated. Protein content, which say specifically it is self-explained for the farming community, progressive farmer, that they should be very careful in application of any supported dose to the crop during application because it can give, sometimes it may harm us instead of defending the crop. This is some statistical correlation. Let me come to the conclusion as per the instruction. The role of the induced biochemical is evident in host plant defense mechanism against the pest. Alteration in this defense secondary metabolite and plant protein may reduce the herbivory infestation and increase the grain yield, number one. The last one is the alteration of this defense biochemical and foliar protein in tested cultivar has been evaluated to utilize the mechanism in IPM approach strategy against leaf folder. Among the 35 tested rice cultivar, Basmati 2000 IR8 shows comparatively best response regarding biochemical and foliar protein alteration against rice leaf folder. So these varieties are recommended for the farming community in Pakistan against the rice leaf folder wherever the population have extensive. So secondly metabolize the phenylalanine ammonia lyase, phenylphenol oxidase, tannine and phenol are induced as a response to rice leaf folder attack. They can be used as a marker for determining resistance in newly developed or developing variety. These enzyme can be used as a good marker in future IPM plan. Nitrogen-based fertilizer should be applied very carefully as it is not only promote negative vegetative growth but also encourage pest infestation by increasing the foliar protein level in the host plant. RM value was increased during the trial. So cultivation of these variety reduce the load of synthetic pesticide and will enhance the fauna in paddy field. The resistant germplasm can be used in a breeding program for development of new high yielding variety against leaf folder. If you allow me, I want to share with the general comments. Okay, there is a gap between the scientist and science and society. By society in this case I mean farming community, progressive farmer, pesticide dealer. Either the scientists fail to deliver the message properly to this community that they should follow the instruction of the laboratory, of the institution, or we fail to convey our message, or they are not convinced with our result, with our recommendation, or there is no incentive for those people to follow the instruction. Like in advanced nation, they have crop insurance plan, so the government have encouraged the farming community to go for crop insurance, and the people trust the government or the state policies, and they get tangible output of the crops. So in this case, you know, as a dean, as a chief editor of Surrey Journal of Agriculture and Journal of Wheat Science, when I see 80% paper I received say efficacy of various pesticide, effect of different chemical. I'm not against any pesticide company or individual. Sorry if I have offended. But no, these are the fact, whatever we are doing. But at least we should now switch to the latest technology and recommendation and new lab technique instead of testing the efficacy. Because you know, in United States of America, if you want to get your pesticide registered or herbicide, they cost $70 million on getting name like Melathion, DDT, whatever. So if they have spent $70 million worldwide, how you can see which one is effective? This is the logic. In our science, in our thesis, we say which one is effective, this is the most. No. Yes, we can use low doses, overdoses, double dose of the tested recommended dose, half of the recommended dose. But don't forget about the efficacy of the pesticide if once it is registered with the commercial company. Therefore, my request would be, so we should follow the instruction of the scientific laboratory, scientist institution to get maximum yield, to keep the population healthy and crop healthy. Thank you very much.
Thank you, Dr. Saab. So, now I will invite for our next presentation. Thank you. Mr. Nasir Qureshi, the President of HOPO Canada. He has expertise in leading transformative information on climate change projects, particularly in agriculture and sustainable development. Mr. Qureshi is also working for poverty alleviation with the collaboration of Sindh Agriculture University, Tandojam. So, Mr. Qureshi sir.
रब्बि इश्रह ली सदरी व यस्सिर ली अमरी वाहलुल उक़दतम मिन लिसानी यफ़क़हू क़ौली. Ladies and gentlemen, distinguished delegates, respected professors and students, Assalamu alaikum warahmatullahi wabarakatuh and good afternoon. It is indeed an honor to be here at this International Conference on Plant Protection. Today, I will be speaking about one of the most pressing challenges of our time, climate change and exploring the carbon management solution in agriculture. Given the limited time, I will be offering a brief overview. However, I will invite all of you to attend our technical sessions where we will explore those critical topics in greater depth. I also warmly welcome you to visit our booth where we are showcasing how farmers, researchers, students are working together in a collaboration on the carbon neutral villages model that is not only helping the environment but also creating unique job and business opportunities. Together, I strongly believe in that together we have a unique opportunity to unite the academia with corporate and the farmers to go against the environment, to fight the to be part of the combat against the environment. How we can do that, we will share you in a moment. No, without further ado, I would invite you to University. Basically, HOPO is a non-profit organization registered in Canada, dedicated to work on a mission to help the deprived communities to become sustainable. Our mission is simple, that how we can help the deprived communities from pity to paycheck and help our supporters to from donation to dividends. With this mission, we are working to redefine the charity. Basically, we are using the United Nations Sustainable Development Goals as a lens to create that environment where we can help the academia, the farmers, the professionals and the researchers to come up with the innovative ways to help those two missions we are working on. One is how to take the deprived communities from pity to paycheck, and how to take the donors, the supporters from donation to dividends. And in this regard, if we look at the environment, climate change is the biggest disruptor of our time. And the climate impact, particularly with the context of Sindh agriculture, huge. But if we look at this, the disaster we are having due to the climate change, meaning of new carbon economy, This is how we are looking at it. And we know that it's a $16 billion agriculture credit market, and where markups goes up to 300%. Agriculture credit is the main source of poverty, and the come up with those. So we are going to do that. Some of the examples are, for example, we are working with the help of the university to launch the biochar moringa pitcher trees. And our objective is, with the help of the university, not only to cure the climate sick soil, but also achieve the target of 1 ton carbon captured in 1 cubic meter. How is that possible? If we can do that as a country, definitely we are contributing to the nature-based solution for the carbon management. So that's the one project we are working with the university. The second project is biogas plant. How we can use the biogas plant to capture the methane, which is 87 times more potent to the carbon dioxide to the global warming. How we can measure that, how we can have a basis where we can benchmark the villages to for the carbon emission, and then step by step neutralize them. So carbon neutral villages is the one major project we have taken with the university to work for in the future. So we are working with the help of university to ensure we are building the capacity of the university in the carbon management. When we are saying capacity building, that means we need the workforce that work in parallel on the carbon literacy agriculture university. We are working how to learn, earn and serve in this horizon, which are interconnected, and how we can come up with the projects where we can educate the second one is academia, the corporate sector, and third one is the farmers and their children. How we can educate them about this carbon management, about this climate change, why there is a global warming, what is happening. So carbon literacy is the main pillar to start this journey. So to start this journey, what we have done, for example, with the help of the university, we have created a survey form using the United Nations adopted carbon calculator. So within 10-15 minutes, when you go to this survey, you answer your lifestyle. When you are living in Europe, your lifestyle is different. When you are living in a developing country, your lifestyle is different. So how much you are contributing to the global warming, you can calculate your own carbon footprint with the investment of only 10-15 minutes. The moment you do that, we award you a carbon neutral bronze certificate. That means at least you know what is your contribution to the global warming. The next natural question would be how you are going to net off your contribution. So in my language, how you are going to clean your environmental shit. So because when we are contributing as an individual, as a business, as a community, then we are responsible to at least take care of whatever we are emitting. So it's all about the education. So the first level is we educate the individual, the corporates, and the businesses, the university, the academia, and everybody should be in this race to at least know what is their footprint, and we issue them the carbon neutral bronze certificate. Now,
सर लंच रेडी है थोड़ी सी अगर समराइज
जी, Again, I'm sorry, due to the time constraint, I have to cut it down. I will just explain this point and then we will go to the next level. Once you know what is your carbon footprint and you have got the bronze certificate, now you have the option as an individual or as a corporate to net off your carbon footprint. For example, if you contribute towards the biogas plant, we will issue you a certificate according to how much you have net off your footprint as an individual or as a corporate. Similarly, if you are, if you have done the 50%, neutralization yourself, we will issue you a silver certificate. If you are you have done 100% of your carbon footprint, you have neutralized that, we will issue you a gold certificate. So with the help of those silver and gold certificate, we have a opportunity for these university students, researchers to earn the money, because the corporates around the globe, they would be their clients and they can do it online. So we train them how to do that. So for that matter, we have launched recently a carbon neutral villages ambassador program. So whoever contribute into that program, he start earning their points, carbon awareness tokens. So as an ambassador or as a student, once you accumulate your carbon awareness tokens, we call them CATs, you have the option to redeem that towards your university fee, maybe your hostel bill, or in worst case, if you need the cash, we pay you the cash. So we have come up with the different products and services with the help of the university to earn the money, so learn, earn, and serve your community. That's the program. So we, at the next session, at the closing ceremony, we would be presenting 100,000 rupees check to one of the students who has been working on this carbon neutral villages, and so far she has earned that money. So everybody has the opportunity to participate that one. Now, because I'm skipping the slides just because of the time constraint, and I would like this opportunity to invite Dr. Manzoor Sahib, Dean of the Crop Protection Faculty, to launch another research project which has been practically initiated by one of my farmer. He took the initiative to look into the Niazbo plant to practically repel the mosquitoes in the Sindh, because everybody knows in the monsoon season in Sindh, mosquito is a huge issue. So how we can have a nature-based, plant which we can use to repel the mosquitoes, which is causing like 30% loss in the milk production and 10 to 30% loss in the farmer income during this monsoon season. So it's a huge challenge, and if we can come up with this mosquito repellent product and services with the help of this Niazbo plant, that would be great. So I'm pleased to launch this research project funded by the HUPO with the university to validate and come up with those products and services. Doctor, please, if you can come and, officially launch this project. Next one.
Dr. Nasir Saab, thank you so much, very much for your kind cooperation with Sindh Agriculture University, Tandojam.
Thank you very much, Nasir Saab. So, due to the time constraint, we only have one presentation, and I request Dr. Atif Jamal to please share your findings in brief with all of us, because we have lunch ready for us. Thank you very much.
Thank you for inviting me. Yeah. So I'll be trying to, I'll be talking about aflatoxin control, and there is a new intervention, that is aflatoxin biocontrol. So I'll be just through biopesticides. So I'll be talking about that. Next, please. I'll skip this. So I'll just talk about aflatoxin, its impact, what is the problem statement, why we want to control aflatoxin, what is the principle of biological control, a success story in maize. This is Aspergillus flavus, you can say the nuisance which is causing aflatoxin. Next, please. So this is the impact that, as there is a saying, health is wealth. So this aflatoxin, unfortunately, is responsible for reducing both these things. Means it affects health, also wealth, because your consignments are compromised. If you send your consignments outside, they are rejected due to the presence of aflatoxin beyond a certain limit. Next, please. So these are the just data to show that there are how many exports are, how many consignments are rejected due to this aflatoxin. And in Europe, you can see that 59.57 of our consignments are rejected due to this aflatoxin. Next, please. So this is very important. There are two phases of aflatoxin, because previously and even now people think that it's a post-harvest problem. It comes in storage. During the storage, conducive conditions are there, and then this aflatoxin is there. But everybody, they just forget about phase one, that how does this aflatoxin contaminate. The Aspergillus flavus is the main fungi which causes aflatoxin. So, and this infection comes at phase one, when the crop is at the production stage or when it's cultivated in the field. So we need this aflatoxin biocontrol. It just focus on controlling the infection of Aspergillus flavus. Next, please. So this is very important. In the morning, this worthy Vice Chancellors talk about this water stress. So this is a study we did that for the first in 2019 and 2022. I collected some samples from same fields in chili growing area, Kundri and Umarkot. And we find out that the aflatoxin contamination was less in 2022 as compared to 2019. As you might know that in 2022 there were floods and there was lots of rain. So we have published this paper that due to stress, this aflatoxin during this floods, and there were more rainfall, water availability was there. So there was no stress for plants. So aflatoxin contamination was less. So it means that this water is very important. And if I could say that this water availability dictates aflatoxin contamination, this is true. So, but the problem is we cannot control the water availability in Sindh. You know that there is rotational water system. So how can we control it? So I'll just go for principle of biocontrol, that we have an alternate. We can use biological control to control aflatoxin. Next, please. So this is also, next please. And then this determining a mycotoxin is very difficult because it's just example of a single year, and you can see that even in the different kernels, the aflatoxin contamination or concentration is different. Next please. This is the lab picture, and you can see that this is ammonia vapor test, toxigenic and atoxigenic stains. The red ones are toxigenic stains, and you can see that there are different, they are showing different variability in the aflatoxin contamination concentration. Next please. So this is the mechanism of biological control. It's simple that we are naturally, this is important to know that the causing agent for aflatoxin is aspergillus flavus. But not all the aspergillus flavus is responsible, or they have, they can cause aflatoxin. There are few natural strains which are called atoxigenic. They don't have the ability to cause aflatoxin. So what happens next please? So this is what countries, these are the area where currently people are using aflatoxin biocontrol. Next please. So this is steps for preparation of biopesticide. You take sample, you test, you need to identify atoxigenic stains, because once you know that this is atoxigenic stain, then you can prepare biopesticide and apply in the field. So you quantify it, you identify, there are 3 methods, morphological, molecular, you check that, and then you apply it through preparation as biopesticide. Next please. This is also, next please. These are different medias used for the identification and processes of aflatoxin biocontrol. Next please. So this is important. There has to be 4, we need to show that there are few characteristics which needs to be fulfilled before we adopt as a atoxigenic stains. It has to be adopted to agroecologically highly competitive. It should be clonal in nature. There should be no vegetative means recombination. Molecular analysis should be, we need to check it that it's really atoxigenic, and it should not produce CPA. Next please. So this is just a picture that how we identify, as I've told you that this toxigenic stains, they show when you apply a ammonia vapor test, the ones which have intermediates, because they produce intermediates during aflatoxin production, so they turn pink or red, and the ones which do not have the capacity to prepare or to make aflatoxin, they remain cream. Next please. So the important thing is why they are atoxigenic. They are atoxigenic because they have some defect in their aflatoxin gene cluster. Fungus Aspergillus flavus has 8 chromosomes, and on the 3rd chromosome there is aflatoxin gene cluster. So so that is the reason the difference between toxigenic and atoxigenic stains is that due to some defect in aflatoxin cluster, they are unable to prepare or make aflatoxin. Next please. Now I'll share you a success story in maize. We, in collaboration with Rafan Maize and USDA, we identify a atoxigenic stain. We named it as Aflapark, and then we apply it in the field. Next please. So these were the field trials. We applied that in 5 districts in Punjab, and we found, next please, that you can see the results that out of 5 fields, 5 districts, 4 they had the aflatoxin concentration less than the control. So that was the heartening result for us, and that is why this Aflapark is being made, and it's now in with DPP for registration process. Next please. Next please. Now presently, because this is a sin, and I have been working for past 3 years in Kunri and Umarkot to identify a few atoxigenic stains so that we can apply it in the field to reduce aflatoxin, which is the main problem. Because as I've told you that in Sindh, this water availability is very important, but it's a rotational system, and we cannot do anything for that. So we are identifying atoxigenics from chilies. Next please. In 2019-20, when I visited Sindh, this area, I collected 52 samples, and you can see that out of 52 samples, more than 38 samples had the aflatoxin concentration more than the regulatory limit, that is 20 parts per billion. Next please. We have identified few atoxigenic stains. This is just a summary. I have summarized it that there are different, based on simple SSR markers, we have identified these strains. Next, please. This is the same. Next, please. So this is just a gel picture of, as I've told you, that there are markers which throughout the aflatoxin gene clusters, and when you do PCR, so you can see that there are different markers which are missing. So, so we identify it and we prepare biopesticide. Next, please. This is just a deletion pattern. Next. So this is a last thing, that before applying it to the field, we do competition assays in the field, in the lab. We apply toxigenic and atoxigenic strains simultaneously, give appropriate conditions to them, and then we check through ELISA or HPLC to see that whether this atoxigenic strain has reduced or not. So, next, please. So these are the results that you can see, that this atoxigenic strains which we have identified, they have reduced up to 96%. So how does they do? Because there is Thigmo response, there is touch system, and somehow the atoxigenic strain, they reduce toxigenic strain. Next, please. Next, please. So this is biopesticide preparation. We do it in the lab and then we apply it in the field. Next, please. So this, you need to see efficacy in the lab, that there is no bacterial contamination, another thing. And then the other last efficacy in the field, it needs to sporulate in the field, because when you apply it as a biopesticide, if it will sporulate, then the sporulation will, this atoxigenic strain will be, proportion of atoxigenic strain will be more in field, and they will outcompete the toxigenic strains. Next, please. These are the future prospects. We need to optimize it for different crops. We need to collaborate with stakeholders. We need to have farmers awareness program, integration with crop and regulatory approval. Next, please. Oh, so this is just the funding poverty, USDA, ALP, I have received funding for that, and all the things which, and my team. So that is what we have done for the last 5 years. Thank you very much.
Thank you very much, sir. Thank you very much, Dr. Saab, for time saving. So now we concluded this session, and I request all the participants, especially the students, that their lunch is available outside this hall, and all our distinguished guests are requested to please join us for their lunch at our rest house. The lunch for students is available at the SAU office. Please collect your lunch boxes from right of this hall.